Thank you for joining us for today's Ipsos expert panel discussion exploring next-generation therapeutics for solid tumors. Throughout today's session, you will remain in listen-only mode. However, throughout the webinar, please submit your questions online using the Q&A feature. Time permitting, we'll be answering questions at the end of today's session. However, if time runs short, then your question will be answered by email. I also encourage you to check out the handouts we've uploaded to the webinar control panel. Today's session is also being recorded and will be directly emailed to you. So now, without further ado, it is my pleasure to welcome today's panel moderator, Joel Sandler, Associate Partner with Ipsos Healthcare Advisory Services. Joel, you have the floor.
Great, thanks, Ellen, and thanks everyone for joining, and those of you tuning in post facto, appreciate it. Good morning, good afternoon, good evening, depending on where you're at. My name is Joel Sandler. As Ellen said, I'm an associate partner with Ipsos, and I work with both pre-commercial and commercial stage therapeutic companies to provide strategic guidance with respect to positioning, partnering, and internal decision making. So would be thrilled to speak to you after the fact if you're interested in learning more. Today, I'm here to talk with an esteemed panel about the topic of next generation modalities in the current and forward-looking era. And so thinking about this in what I call the post-PD-1 world, that's not to be conflated with therapies that can be applied in the post-PD-1 setting, but rather the fact that checkpoint inhibitors have made a huge impact across various solid tumors, and yet there's plenty of opportunity to improve upon the current standards of care across these different tumor types. And that value proposition can take on many different flavors. And so to that point, I think the discussion today is going to be touching upon a number of different modalities, ways in which they could potentially be positioned for success in the forward-looking competitive landscape across different solid tumor types, and what that can mean for patients. And so let me just, before I get to my panel, and we're going to have a great discussion in the next hour, I just wanted to share some initial thoughts as well as some original data that we generated in preparation for this discussion. So as I said, the question is, what are those gaps that remain? What is the promise and limitations of different modalities in solid tumors? Okay, so 2024 was a good year from the perspective of those tracking FDA regulatory approvals. In oncology, we saw 62. That's up from 45, and even lower numbers in kind of the COVID dip. So we do see a lot of activity in terms of regulatory approvals, just spread out across a number of different modalities. To be clear, some of these are line extensions, and some of them are not necessarily going to be novel chemical or biological entities. But in the main, I think we did see a number of different interesting modalities. And so we went out and we we surveyed medical oncologists and asked them a few questions. First and foremost, based on these 62 FDA approvals in 24, which of the following modality categories will have the greatest impact on solid tumor patients? And surprisingly, to me at least, the top answer was bispecifics. Now, we know there were a couple of bispecifics that were approved in Imdeltra, for small cell lung. But it's not the lion's share of approvals, and yet I think what the oncologists are telling us here is that these next-gen modalities like bispecifics, like IO, there's been, I would say, pent-up demand given everything that's happening on the hem-onc side of the divide. And so now that we're finally starting to see some cell therapy approval, some T-cell, some bifunctional biologics in this space, in addition to antibody drug conjugates, which I think everyone's excited about, that's really, I think, what has their antennae up. So just kind of looking back a little bit, so Eric Blauen here at Ipsos has surveyed oncologists now every year for the past couple of years, and we'll continue doing this, in terms of what they see as their favorite modalities, those that hold the greatest promise. Now, this is not specific to solid tumors, but I think it does underscore the point that things like antibody drug conjugates are coming up in terms of interest level. And in 24 versus 23, we subdivided between those delivering novel payloads, like degraders and radioligands, versus those that deliver more kind of classic potent cytotoxics that you would expect to see from an ADC. And so the interest in those is coming up at the expense of bispecifics and arguably maybe other IO immune checkpoint inhibitors. So some interesting dynamics there. When we double click into this and ask the question, all right, you know, if you had to pick your top 3 favorite modalities, Mr. or Mrs. Oncologist, what are those? And again, kind of coming back to the bispecifics, the multispecifics really capture the most, at least in terms of being ranked first, followed by ADCs with novel payloads, and then T-cell therapies and some other modalities behind them. Asking the same pool of oncologists, what do they find as the most impactful events that took place in 2024? I thought this was really interesting. By far, the most popular was Enhertu's tumor agnostic label. And you can imagine from a utility standpoint, that does become very attractive for patients that are in that heavily pretreated setting. You're not really sure what to do with them. Having that tumor agnostic label is very helpful. And so that's what we're hearing. Iovance finally got their TIL therapy approved. And so I think that's something that had been anticipated for a long time, the fact that it's up there along with Imdelltra, which I mentioned. And then only when you get down to the 4th and 5th favorite options do you get to things like VEGF or PD-1 bispecifics and pan-RAS inhibition. So these are some areas that I think investors were very excited about. But when you talk to oncologists, it's a bit of a different story. So again, interesting data. And then finally, you know, I just wanted to make sure that we're not forgetting about some of the more tried and true workhorses of oncology, the small molecules. And in the kind of, you know, usual run up to JP Morgan, where you see a couple of different acquisitions, 2 of those were bolt-on acquisitions, one by Lilly, another by GSK, both in the targeted small molecule space and both with respect to solid tumors. So I think that there is interest, at least on the partner side, in terms of opportunities where there's a clear return on investment. You can kind of, you know, quantify the patient population. You can carve out subsets of larger heterogeneous tumor types, and you can do so with relatively high confidence that you're going to be able to move the needle and perhaps leapfrog into earlier lines of treatment that you might not be able to do if you're taking more of an all-comers approach. All right, so those are just some initial thoughts. I'm gonna stop sharing and we'll go to panel mode, and I will ask each of my esteemed panelists to quickly self-introduce themselves, the companies that they're representing, the modalities, and as well as some initial thoughts before we get until we get into discussion topics. So Randy, I see you at the top of my screen, so maybe I'll ask you to start.
Hey, Joel, thank you, and great to be here. Thank you, and looking forward to a great panel. It's really an honor to be part of this. So I'm Dr. Randy Heyer. I'm a physician, a scientist, and a storyteller, and I am developing with Merlin Biotech, that's my company, a messenger RNA encoded natural immune target. This is a deubiquitinase, so it removes ubiquitin from other proteins in a specific pattern. And this one, number 6, has a very interesting immunostimulatory pattern that we see we certainly see the effects of that in vitro and vivo data and complementary human epidemiologic data. USP6 as a therapeutic could have potential to be one of those tumor agnostic style therapies. Time will tell, of course. And given that you want to be short here, I'll end now and hand it to the next person. Thank you.
Thanks. Mark?
Hi, my name is Mark Froelich. I'm a medical oncologist by training and been doing immuno-oncology and specifically cell therapy for most of my career, for the better part of 25 years. Excite therapies, Dendrion, Juno Therapeutics, Pact Pharma, and most recently Adaptive Therapeutics. And I would say in terms of the question that you've brought us together for here, Joel, I'd say that, I think it's been a little frustrating, the progress in solid tumors. I think we all thought once the checkpoints got approved that we just had to find that second, magic bullet that we could add to checkpoint inhibition that would really make a difference in solid tumors. And I think the progress has been much, much slower than we had hoped. And we saw a lot of, I think, disappointments with things like, the IDO pathway, adenosine, CD47, TIGIT, novel checkpoints. LAG-3 was kind of a base hit, but not a home run. And so I think the real question is, is there a reason to be optimistic here? And I think what we'll talk about today is hopefully, I think there are emerging evidence that, things are starting to work in solid tumors. Specifically, what we at Adapt are working on are adoptive, off-the-shelf natural killer cell therapy. And I think one of the real attractions about cellular therapy is the ability to really multiplex multiple different mechanisms within a single therapy or product. And specifically, I think one of the attractions about, innate cells like natural killer cells or gamma delta T cells are that they have multiple different sensing mechanisms and receptors that help them distinguish between normal and abnormal tissue. And so the ability to kind of provide that specificity and ability to kill where there's multiple different tumor escape mechanisms, I think is real promise of these therapies. And specifically, we've identified a subset of natural killer cells that are extremely potent, that are epigenetically modified in response to exposure to cytomegalovirus. And we've shown preclinically and now early evidence of clinical data in the setting of myeloma that these cells are in fact very active. And so our goal is to really validate the platform in the heme space where cell therapies have been more efficacious, and then in the future turn our attention to solid tumors. Thank you.
Great. Jeff.
Thanks, Joel. My name is Jeff Toll. I am here representing Novartis today, and specifically radioligand therapies, which are a way of delivering radiation ultimately directly to the cancer cells. And really the promise here is that we get to build on two proven therapeutic paradigms. First, what Joel just described in terms of even what's being done within small molecules, the being able to build on the precision of the targeted therapy revolution, right? Leveraging the molecular understanding of cancer biology to selectively home in on specific proteins or pathways that drive tumor growth. But with an RLT, what we then do is attach radiation or radioisotope to that as a payload, which of course, radiation's been a cornerstone of the treatment of cancer now for, probably over a century at this point. And is incredibly effective at destroying cancer cells, but compared with external beam, you can use RLTs to ultimately target cells wherever they may exist in the body. Then because we're using ligands that have both high specificity and affinity for the tumor-specific receptors, the unbound molecules are rapidly metabolized, which results in a great safety profile for these drugs as well. So really what we're trying to do at Novartis, we have approved therapies in both neuroendocrine tumors as well as advanced stages of prostate cancer at this point in time. And the promise we're trying to build on here is to bring these across a variety of different solid tumors in the future.
Regan?
Hey, I'm Regan Jarvis. I'm the co-founder and CEO at Anoka. We're a T-cell biology company based in Sweden. The company's built around a unique cell biology platform that allows us to systematically map in a quantitative way the HLA peptide targets of T-cells, generate and characterize the T-cell receptors, the cognate T-cell receptors to those targets, and even do functional optimization of TCR frameworks, all in a cell biology context, which is really important to understanding the fundamentals of both target presentation and T-cell activity. We've primarily deployed that platform towards TCR T-cell therapies in the oncology setting. and constructed a very deep pipeline, because that's the name of the game in the space, is scaling out across this very rich tumor selective target space. Built internally a manufacturing facility, deployed in there a non-viral gene editing methodology, which has some advantages in the TCR-T space with dealing with endogenous T cell receptor genes and improving quality, but also allows you to scale out more rapidly and cost effectively across those targets. First clinical program will start enrolling first half of this year, will be a KRAS, or mutant KRAS targeting TCR-T product. And really look forward to the discussion today.
And Kai.
Yes, thank you very much. Hello from my side. So I guess I'm the youngest panelist here, but we are developing one of the oldest, most already established therapies on the market, maybe developing a, let's say, more precise form of chemotherapy. So I'm a trained molecular biologist, now in the role as business director for NAP-CTX. We are a German company, and we aim to redefine the safety and precision of chemotherapy while retaining or maybe even improving its well-known efficacy. So we develop a platform approach that basically encapsulates platinum-based chemotherapeutics in an inactive form. And these encapsulated nanoparticles that form then are later activated via ultrasound. So in Germany, we like it precise and maybe punctual as well. And we can then activate our nanoparticles when we want and where we want. And I guess this is quite important, because chemotherapy will be on the market, will be important for the next years, definitely. But they come along with a bit of a side effects, and especially for the patients, it's devastating. So we hope to really minimize, thereby minimizing side effects for the patients, but otherwise betting on an already well-established therapy.
Perfect. So, I'm very, very proud of this, the way this panel has been curated. I think we're able to speak across, and between us, across the breadth of different modalities that are being brought to bear in the forward-looking solid tumor landscape, from cell therapies, which are well represented both on the innate and adaptive side of the immune system, between Mark and Regan, targeted therapies, thanks to Jeff being able to speak to radioligand therapies, Randy being able to talk about the ability to augment and perhaps address colder tumors or those that are warm to make them hot. And then Kai, who's really, along with probably some other companies out there, who's betting on the value of chemotherapy and being able to retain the benefits while improving upon some of the liabilities. And so I'm really excited for the discussion. So let me start with, I think, an area that I think Mark alluded to, and I think it's the right place to start, which is that idea that held people in thrall for a couple of years after CTLA, and certainly after PD-1 inhibitors demonstrated responses in heavily pretreated cancers and durable remissions, at least in some of those cases. And that's really, I think, why people get excited about immunotherapy, the ability to bring the host immune response as an ally, as a weapon against cancer, which is a very difficult target. And so there was this flurry of activity, I would say there still is, in terms of different modalities that could be combined with checkpoint inhibitors and take us from really good to even better. And I think what we saw was in hindsight, perhaps predictably so, a bit of disappointment in terms of the ability to further unlock this pathway. And so maybe, Randy, if you could just start us off on the topic of, and I know, given that Merlin is focused on vaccines, maybe more broadly speaking, this idea of being able to augment, improve upon the checkpoint inhibitors, which have done well, but I think have also given us clear paths to improve upon. But nonetheless, it's been a challenge for the industry.
Sure, certainly. No, thank you, Joel. I mean, I think first of all, Merlin's product is a little bit more than a vaccine. It does use the mRNA technology, but I think it has a vaccine-like effect, but it's broader, and I'll be talking about that. And I think your point about making cold tumors hot is one of the things that the deubiquitinase really does do, and I think that's important, obviously, in immune therapy. So to answer your broader question, I think listen, as a physician or patients, I mean, immune checkpoint inhibitors have been fantastic since their introduction in 2011. When they work, they're great. They really do. I know personal friends of mine, some younger than me, who've unfortunately had cancer, and they're living, breathing, talking to me because of immune checkpoint therapy, and that's real. So, hats off. 2011, they came out, and then there was I looked at an ASCO abstract from last year. The response rates initially was estimated pretty meager, 0.14% overall, of course, and then as high and rising to 19.6 in 2023, which of course represents a very nice trend and better targeting, better use of these important medicines. Nonetheless, they have their shortcomings. I think we all know what they are. They're not to mention the low response rates, but there's also, of course, there can be some pronounced toxicity. And in clinical studies that I've overseen, we had more than one dose-limiting therapy and treatment-ending adverse effect. So it's real. It's very real, and it's something that wouldn't come as a huge surprise when you have a medicine that's so powerfully able to activate the immune system. And so, how do we build on that and go forward? And I think, Joel, you hit, I think this, first of all, immunotherapy is great. I mean, that's what the body is doing anyway every day. So why not just augment that ability in the face of a cancer that's threatening our life or certainly our livelihood? And I think there's lots of great, I'm really privileged to be here with everyone else because they have such, nice angles on this. And I'm not sure any one angle is going to be, the angle, and maybe they all work together. We'll find out. I can talk briefly about what I see the value in the product that Merlin is trying to develop, the deubiquitinase, and how it fits into this. And the deubiquitinase does have a broad effect on activating the immune system. It increases the interferon gamma-like signature, turning those cold tumors hot. It increases cell surface ligand expression. It activates other immune cells, including natural killers, producing quite a potent immune environment in what's otherwise potentially a cold tumor. And also, we do have data that shows that we see not only an innate response, but an adaptive response. So, the deubiquitinase is making the immune system recognize some of the tumor neoantigen, producing adaptive responses. So it's quite potent. And that medicine right now, we call it MER-101, is preclinical stage. Will be in the clinic next year. And of course, I see combinatorial approaches, of course, and that's the mainstay of cancer therapy. There's actually data right now, published data, looking at people who are on immune checkpoint inhibitors, both CTLA-4 and PD-L1, with melanoma, and you can measure USP6 levels in people because it's a natural protein. We all make it. And it turns out that people who naturally have high levels simply live longer than those who naturally have low levels, and actually bifurcates rather nicely. And we do see that effect epidemiologically in a number of other tumors, epi data, with some companion in vitro and in vivo data supporting activity across tumors from all three primordial germlines, so suggesting clearly a higher order actor. Now, this hopefully will prove itself out in the clinic and become an important tool. I think it may be a tool by itself. It will certainly be a tool, a combinatorial tool, perhaps with many of the other therapies being discussed today. So I'll leave it at that for now because I want to hear from the other panelists. I'd like to continue to participate in this important discussion. Thank you.
Yeah, maybe Mark, since you kind of brought us here in the first place, I mean, we saw, to the point about cold and hot tumors, it's such a compelling idea, right? And an intuitive one. But I think it's also telling that Genentech shuttered their immuno-oncology in an R&D reorg. And they were kind of the original purveyors between Chen and Melman and some of that noise making, at least on the industry side, I think they were really telling that story about subdividing and addressing cold tumors. So what happened there, and kind of how do we learn from lessons going forward?
Yeah, no, for sure. I think it's sobering, right? It's been a decade and a half since these approvals, and we just haven't found that silver bullet yet. But I think there is reason to be optimistic. I mean, I think specifically just to riff off, Randy's discussion here about cancer vaccines, having spent 8.5 years, developing Provenge, which was the first therapeutic vaccine approved back in 2010. It's been 15 years. There has not been another therapeutic cancer vaccine approved. But the reason for optimism, right? I mean, I think there's the recent, randomized data in melanoma, BioNTech, Moderna, showing that these personalized cell vaccines, neoantigen predicted, are really improving progression-free and overall survival. And I think it's gotten them enthusiastic enough to kind of foray into other solid tumors like non-small cell lung cancer. So, and I think, other approaches like Randy discussed, Nuvagen, a company that I'm involved with, using kind of off-the-shelf approaches that don't require you to predict what the neoepitopes are, I think hold a lot of promise for other settings like the neoadjuvant setting, for example, that are more challenging to do. So, I think there is reason to be optimistic about, whether it's cancer vaccines or the other fields that our other panelists are involved here. I think finally now we're starting to see hints of efficacy, and I think that's what we can really build on, is like understanding it, where it's working and why it's working, and leverage those specific indications or applications to hopefully be more broadly applicable across other tumors for more patients.
Yeah. I do want to get to cell therapies because I think there's kind of a natural transition there, but I don't want to negate the fact that cytotoxic therapies, targeted therapies, radioligand therapies, platinum-containing drugs have immunogenic effects. So, Jeff or Kai, I don't know if either of you just kind of want to weigh into that point and the fact that maybe the distinction between targeted and immunotherapy is a little bit fuzzy, at least.
Sorry, I'm happy to jump in here. I wasn't sure if Kai, you were going to do it, but
go, Jeff.
I mean, it's interesting because I think, of course, the interest in immuno-oncology has prompted, I think, everyone to figure out a way to describe their therapies as having some sort of immune activation activity or synergistic, et cetera, or certainly at least to have that exploration, right? And so I think, whether we're talking about radioligand therapies or otherwise, I think there's a lot of opportunity, and you're seeing that with all the combination work that's being done, whether it's with ADCs, for example, or other modalities to combine with checkpoint inhibition because of the complementary mechanisms and representing RLT. I think in a very similar way, I think it's possible we'll discover that may also be a synergistic combination. There's some early data showing that, with the use of RLTs, you can induce antigen release that may increase sensitivity to, these checkpoint inhibition or other immuno-oncology types of drugs. And conversely, of course, that immuno-oncology agents may also provide radiosensitization to tumors. But, it's still a very evolving field and something, certainly we're going to be exploring. And I think that's probably true for a number of agents. And I know with Kai, the role of chemotherapy as being both synergistic as well as having, immune stimulated activity as well.
Yeah, absolutely. So I do not have so much to add, but of course, for chemotherapy, I mean, already you can use it neoadjuvant, you can use it adjuvant, palliative, and for example, when we look at PD-1, you have the problem with cold tumors. And with chemotherapy, I mean, the approach of chemotherapy, the mechanism of action, it is quite simple. You always are able to destroy, to kill the cells. So you release pumps and dumps that activate sometimes more, sometimes less the immune system. So you can, of course, combine it, chemotherapy followed by immunotherapy. And it works. It works not in every cancer type, but in most of it, or we will see maybe in most of it. In some it works. I mean, for example, in non-small cell lung cancer, it was able to increase, I think, the 5-year survival of the patients from 20% to 40% by just combining these two therapies. And I think this is also the way to go in the future. So coming away from thinking, hey, we have one therapy approach and this works for this type of cancer and this type of, I don't know, maybe biomarker specified patient population, but rather combine the different therapies. We have to see what makes most sense, also in which order, of course. But the direction for immunotherapies needs something like a chemotherapy to transform a cold tumor into a hot tumor, for example.
Yeah. Okay, great. Now, for cell therapies and immune redirecting biologics, I think we can add to the list. We saw in the survey data, Regan, that's what the oncologists are most excited about. I would imagine, investors, there's some skepticism, at least in terms of given the cost, the commercial complexity. But I think what we're hearing is, excitement that you can, obviously from hematological malignancies like multiple myeloma and non-Hodgkin's, but I think now there's a lot of breakthroughs happening in almost on a weekly basis with these modalities in solid tumors. Can you just maybe bring us up to speed on the state of play? Where are we with cell therapies in solid tumors?
Yeah, I mean, obviously we're chasing the success of CAR Ts and to a lesser extent the T-cell engagers, let's say in hem-onc, right? And that's it's a target game. There's some great targets in the hem-onc setting, you have BCMA, CD19, and replicating that success in solid tumors is going to require targets. I mean, you saw TIL from Iovance get approved last year, which is not really a target specific one, but it is a solid product. And then you saw the TCR-T out of Adaptimmune, which is chasing this sort of not unique, but a novel in terms of therapeutic modality target space, which is tumor selective. This is the real challenge for CAR Ts and T-cell engagers. In fact, ADCs and radioligands as well in solid tumors is what is the tumor selective, the tumor restricted target that you're going to chase. So I think that's really where the field is going. And of course, I'm slightly biased as running a TCR-T company. I think those HLA restricted peptides are the targets to be chasing. There's a huge caveat there, of course, which is this is a highly segmented target space. So you need to scale out, almost personalize these therapeutics. And that's going to, therein lies the challenge. How do you do that discovery, that target mapping, the asset development, and of course the product development, even the manufacturing in a scalable way? But I think that's the direction of travel. Also the interesting data already mentioned on the vaccines in solid tumors as well, which is all that, bring that together. T-cell responses are going to drive, I think, at least the initial responses in solid tumors that we need to build upon. It was already mentioned that there's probably not magic bullets here or silver bullets. All this is going to work in combination, either molecules, vaccination with cell therapies, or just other combos. So I think it's all these targeted therapies need targets, and that's the challenge in solid tumors.
Mm-hmm. Yeah. Anyone else want to weigh in? Mark, I know you've kind of
sure. I think as we talked about, it's taken a long time for there to be hints of efficacy in the solid tumor setting. And in addition to the ones already mentioned, I think BioNTech has some interesting data with their Claudin-6 CAR showing efficacy in germ cell and ovarian tumors. There's been some case reports of, in brain tumors with EGFR and IL-13 receptor alpha 2 showing efficacy. In the TCR space, there's a New England Journal report from Eric Tran on KRAS that showed some, pretty impressive responses. Although I think when they looked at a larger number of patients, it wasn't, clear what percentage of patients might benefit from that. HPV, Christian Heinrich's data, there with the TCR showing a benefit. And then in the TIL space, that's been initially just shown in melanoma, but I think Iovance also has data in the kind of the non-small cell lung and ovarian cancer setting. So, I think there are hints now that these are starting to work in solid tumors, and I think that's highly encouraging. I think the other area that to address kind of the logistical and cost challenges is to use kind of off-the-shelf allo cells. And a number of folks are doing that, principally starting in the heme space, Allogene, Caribou, CRISPR, and others. And, I think those early data are encouraging, and it looks like, the 6-month CRs in some of those studies are looking comparable to the approved products. So, I think those will definitely make a foray. And then obviously, I think these other cell types, as I said in my intro, I think the innate cell types like natural killer cells, gamma delta, NKT cells, macrophages, like Charisma is doing, I think may have some ability to kind of home and address the tumor microenvironment perhaps in a different way than a T cell does. And I think those are highly encouraging. And then finally, I think it is worth talking about, because I think your respondents, highlighted bispecific engagers as really promising, the T cell engagers, and those are, approved, as you noted, myeloma, lymphoma, very encouraging. But now I think we're starting to see, evidence in solid tumors as well. So the DLL3 from Amgen and small cell lung cancer, and then just recently, in prostate cancer, both Janux and Vir have, against PSMA, have showed some really encouraging data in a tumor that, is felt to be very inhospitable, kind of a cold tumor. And, checkpoints haven't worked there, and unclear why. But I think they're seeing PSA, major PSA responses in the majority of patients from both those data sets. That suggests to me that these are, obviously that's not an approvable endpoint, but will likely translate to, PFS or OS. And I think, that to see traction in a tumor like that, again, historically is not felt to be responsive, I think is reason to be optimistic.
Good. Randy, I have a kind of a question, Joel, if you don't mind. Is that, I'm thinking, one of the things that's really powerful learning technique is what can we learn from failures in immunotherapy that guide our development programs and our targets forward? And I'd be interested to see what the thoughts are of my panel members, or you, Joel, or anyone.
Yeah. I think, well, just an initial thought on that is, there's so much optionality in terms of, what to combine with what. I think there's also a question about expectations, whenever there's a big breakthrough, kind of a sea change, and you go from here to here with a modality. We've seen historically that that's where competitive crowding rushes in. That's where a lot of dollars are spent,
right,
because there's a belief if you can go from here to here, well, how much harder is it to go here? And what we learn is that biology is complicated. So that's kind of my initial answer to that question is there's a lot to think about here. But to Mark's point, and back to you, Randy, about how we're finally seeing a vaccine story coming to fruition, potentially in the oncology space after all these years post-Endrion. There's a lot to learn, right? And there are a lot of failures. And I think that there are some very smart people out there, including yourselves, who are, harnessing the lessons of the field and kind of coming back. And there's probably also a lot of investigator learning in terms of what the endpoints are to be looking at, the surrogates that kind of give you a sense that there is reason to believe and reason to kind of pursue a given direction.
Yeah, my thought on that is, if you look at all of those high-profile failures I mentioned at the beginning, right? IDO, adenosine, CD47, TIGIT, these other checkpoints. I think part of the challenge is that, as you noted, biology is really complex, and there's redundancy in these pathways, right? You target one of those in the adenosine pathway, but there's another receptor, right? And so I think I think we probably, as a field, jumped into some of these early before we fully understand kind of those redundant pathways and how to target some of them simultaneously. And I think some of those targets may come back when we're a little smarter about figuring out how to address that redundancy.
Yeah, good question. I do want to kind of shift. We were starting to talk about prostate cancer, and Jeff, I know you guys have jumped in with both feet in radioligand therapy. So maybe you could tell us a little bit about that decision. How to potentially get around some of the hurdles with this novel modality, and then more broadly, and this is maybe for the panel in general, this idea that you can go after the same tumor-associated antigen with a different payload or a different modality and somehow achieve differential clinical benefit, right? We've seen that with antibody drug conjugates. And it's a lot of the reason why, if there's a validated target from a monoclonal antibody, people will take that same target with a cell therapy and a bispecific. So Jeff, I'm not asking you to weigh in on all of that, but I think maybe you could start with RLTs, what success looks like there, what are some of the hurdles, and then maybe we can open it up more broadly.
Yeah, sure. Happy to kick that off. And with RLTs and full transparency, I was not with Novartis at the time that they made the decision to move into this space or to acquire the various companies that were working in it, but joined a couple of years ago. And really, I think the premise here is that, as you said, right, you've got PSMA that's well expressed in prostate cancer cells, and so in some way, an ideal target for this concept of taking a targeting ligand and then attaching some sort of payload to that, right? And so you're seeing that with T-cell engagers now, as others have mentioned, as in the same way that we've done it with RLTs here. I think that part of the challenge, of course, in developing in oncology is ideally you want to be able to move earlier. And I think that's one of the things we think is promising with radioligand therapies is the ability to, in patients with much earlier stages of disease, potentially, use the power of radiation, but use it on systemic disease in order to reduce the risk of recurrence. But of course, in oncology development, you've got to start really late, which is where our therapy was initially approved. We hope for an approval in the not so distant future in a somewhat earlier setting and still in metastatic castration-resistant prostate cancer. But I think one of the fun things about RLTs as well is that these are essentially a little bit like Legos in the sense that you get to swap in and out different components based on what you want to do, right? And so, in the same way that our current therapy is several components, right? You have a ligand, a chelator, a linker, and then you have your radioisotope. You can swap that radioisotope out, which we've now done for a therapy that we've announced is entering phase 3 in prostate cancer as well, switching out the beta emitter for an alpha emitter as well, which gives you a different response profile, different wavelength and radioactivity that we think will have a differential profile within prostate cancer as well. You can also swap out the ligand to go after either different stages of the disease potentially based on the expression profile and its specificity to it in either earlier or later stages or in different tumors entirely. And that's one of the things that we're looking at as well. So maybe I'll stop there and see where you want to take things, Joel.
Yeah, no, well, I want to take it back to Regan, because Regan, you talked about targeted, the need to be targeted with cell therapies, and obviously I think TCR T cells are the epitome of that. But just kind of the idea that the target is a big deal, and just because a target produces suboptimal effects in the context of one modality, like Jeff, we saw PSMA for a long time kind of languish in the clinic, I think, before you attached a radio or someone attached a radioligand to it and generated approvable responses. So Regan, maybe you can talk about targetability, the value of the target, and how you can potentially get a bit of a boost with a novel modality against the same target that others have tried.
I mean, sometimes it's not about the modality. I mean, different ADC, different chemistry, different radioligand. Sometimes, because it's such a low volume game, it's just the luck of the PK/PD or how good your team is in driving the right type of profile of your drug. At the end of the day, if you look at, let's say, cell therapy examples, generally speaking, the CAR Ts have better on the same targets than the T-cell engagers, for example. By not a huge margin in some instances, but you're cutting out the middleman in a way. You're just reprogramming that T-cell to go in and kill the malignant cells, right? I think it's a much, as I alluded to earlier, a much tougher game in the solid tumor setting, where the number of tumor selective or malignant cell selective targets is, say, fewer or less obvious than, say, a CD19 or a BCMA in the hem-onc setting. And they're much lower expression if you're talking about the PHLA targets for a TCR-T. So then you look at the head-to-head, a lot of it unpublished and very early stage, but TCR-T versus the TCR mimetic, engages like an Immunocore product, for example. And you're seeing, higher efficacy driven by the cell product rather than the biologic. And I think that's just owing to the inherent sensitivity of T cells. You program a targeting receptor, especially a TCR, and you just drive a much more sensitive response against these very low expression targets. And some inherent biology there as well, where these are very subtle, again, talking about the HLA peptide targets, these are very subtle molecular presentation. This peptide is, I often say, a sausage in a bun, but it's really like a little meatball in a giant bun, where you've got this tiny little peptide to try and contact with a biologic, even a recombinant TCR. And it's very hard to drive the sensitivity, the affinity in that case, without driving off-target effect, without driving promiscuity. And it's something a T cell can do, because that's what T cells evolved to do, of course. So on the drug side of things, it's not always about the modality. It's sometimes, good luck, bad luck, good program, early stage to get the results you want to move forward with. But I think inherently, if you're reprogramming T cells with the right kind of receptor against the right kind of target, you're jumping off at a better position than trying to engineer a biologic to do the same thing.
Yeah, especially if you're talking about patients who are kind of in that pre-treated setting, their own tissue may not be in the best shape. And so either taking their own cells and kind of, boosting them ex vivo, or an allogeneic approach like Mark alluded to earlier. Okay, other comments, and this is maybe just kind of a broader question about target versus modality. One thought I always have is, is the expression profile of a target in terms of on versus off tumor expression, is that the most important thing? Is it the function of the target? Is it some combination thereof? And, I think these are always kind of empirically derived. We don't know. You can't always kind of assume, but in the case of TCRs, Regan, we see Immatics data. It's very, very compelling with PRAME. And so I think that there is something to be said for a hyperselective or selectively expressed target, but I don't know if anyone else wants to weigh in on that.
Well, I'll just make one comment, Joel. I want to pick up on Reagan's comment. I thought it was pretty clever. If you have a targeted therapy, you need a target. And that target may shift, may move. Cancer is pretty clever, right? It got to where it is by outwitting the immune system. So that's something that shouldn't be lost on us. And so in the target environment, that's something we really have to think about. And I do think this is where the chemo still is part of the story, because chemo may not require so much of that, and especially in a combinatorial approach. And I think this is a real attractive, pattern forward. I also want to just mention briefly that I'm certainly a big fan of messenger RNA because that platform has been de-risked. We know what it does. We know how it works. We can tweak it. We can do all kinds of things. I think it's just opening up. I think the days are early. And not just the personalized cancer vaccine, which of course looks extremely promising. Time will tell. But there's many others opportunities open there that I think some of them, I'm not even sure we've conceived them yet. So I think that's a sort of a if you want to call it if COVID had gifts, that may be one of them. And so that's I wanted to key on that point that I thought Reagan said that rather well. And
it was definitely a gift to companies like Moderna, who I think were looking for a home for that modality, and they sure found it.
Oh yeah, and that modality has been de-risked. It's heavily de-risked now. It's a non-issue, and we now what we can do with it.
Yeah, but I want to pick up on your first point, which was the need to have a target that isn't necessarily going to go away, or that you're not going to select against with a hyper-targeted therapy, and maybe with a TCR, you know, and some sort of target that's involved in differentiation, you know, like a testis antigen target, that's less of a concern. But with overexpressed targets, that can be an issue. And so the need to target pathways, or you know, so in your case, Randy, you guys are targeting the ubiquitin tagging pathway, which is something that I would imagine cancer is going to rely upon, and there isn't going to be a lot of redundancy. We only have a few minutes left, but Kai, I want to make sure that we didn't neglect to talk about the chemo story, and obviously you guys are, you know, betting that there's value there, and I don't think anyone's going to argue with that. But on the other hand, your job is challenging because, you know, there are a lot of different directions you can go with it, and you have to demonstrate value against a generic standard, you know, benchmark. So maybe talk a little bit about that and what success looks like for you guys.
Yeah, absolutely. Thank you. I mean, in regards to target, maybe the smartest target is that you have no target that can somehow evade or be deleted by a mutation or whatever. And I think, but this is, so let me explain what we do is that we have a targeted approach that does not depend on a molecular target, let's say, but we are using the so-called EPR effect, enhanced permeability and retention effect, which was described first in the 80s. And it's quite simple because it states, and it was shown now for years, that small particles in the nano area, they are able to accumulate, so first target and then accumulate in tumor tissue because of the size of the particle itself, and on the other hand, because of the characteristics of tumor tissue being not as dense as healthy tissue. And that is what our approach does and relies on. So it's a passive accumulation effect, but we come along without having any specific antibody that we need, without any biomarker that we have to look for. And I think in terms of chemotherapy, that this makes sense, of course, but I guess also for other approaches, it would make sense to have a look at that. You can combine this effect with several other modalities like mRNA targeting and so on. In our case, so we believe that there is no way around chemotherapy also in the future. When you see predictions, the market is basically predicted to double in the next 10 years to almost $20 billion on a global scale, of course. So meaning it will be used on patients, but if you ask patients about chemotherapy, they are afraid, they don't want to have it. I mean, it's very efficient, but the side effects are detrimental. Meaning that if you can just change this from a systemic approach to a targeted approach, we already know that it works. So this would be the major step with it. And what comes along with it is that you then get rid not only of the toxicity, which is the first problem, but you also have the second problem, not so common, is the resistance to chemotherapy that happens. And also here we think that when you have the chemotherapeutic in a higher concentration brought to the tumor, there's a bigger chance that you overcome also the resistance that takes place at some point at least. So this is what we bet on. This is what we hope for, that we can prove it. We are quite early on, but we have some promising results, let's say, although only in animals. But we hope to bring it to the patients. So 30% of patients are discontinuing chemotherapy because of the side effects. Would be great to have the same efficacy without having these side effects and taking away the patient's fear for this therapy.
Yeah. All right. So you have a worldview of a future in which chemotherapy still plays a role, but we can do better without necessarily getting rid of it altogether. I think if you're Mark or if you're Regan, you have a worldview where cell therapies play an active role across the solid tumor landscape. Randy, I think, and I don't want to put, well, I guess I am putting words in people's mouths just because we only have a few minutes left. But, the ability to augment and perhaps address, kind of in the cracks of areas where immunotherapy has thus far fallen short, as well as, fundamental pathways that need to be, that cancer relies upon, and somehow shutting those down, or at least using that as a hook to get a targeted immune effect. And then Jeff, radioligand therapies. I mean, I think that's incredibly exciting, the data that Novartis has generated. It's not for the faint of heart, so I think you guys have some barriers to entry amongst your competition, so that's also a good thing. So 2 minutes left. I'll invite anyone if they want to just kind of jump in the fray with some final thoughts, and don't be shy.
I mean, just in terms of even barriers to entry with RLT and whatnot, I mean, it is a very different universe. It's been interesting to get into this space over the last couple of years, and the obvious stuff being, these drugs have half-lives, and thus everything from sourcing, manufacturing, distribution, ordering is completely different than how most of this works, and that, your product is decaying the entire way. The radioisotopes are expensive, all sorts of fun stuff. So certainly, it's a really fascinating area to get involved with and to learn a lot about, and hopefully we can apply that to even more tumors in the future.
That's great. Yeah. I think barriers to entry is the name of the game. We see that with cell therapies too, right? I think if you can, or owning a piece of biology or an understanding of technology. Anyway, so I think with that, I'm gonna. Sorry, Randy, did you want to?
Got my last parting shot, yeah. So no, just real briefly, I think one of the challenges, I think Joel may have said it, or somebody said it well earlier, is that one of the challenges, of course, is that investors aren't necessarily, they tend to cling on to something that has traction either in the press or, and certainly in the approval pathways. And, and sometimes that isn't the most efficient use of capital, and it, capital is diverted, attention is diverted away from things that really could go places. Now, I don't have a crystal ball. It's just that it is a phenomenon, and I don't know what the answer to that is. It's just definitely out there.
All right. Well, with that, we are at 2 o'clock Eastern. So thank you to my panel. Thanks to all of you who tuned in, and we'll catch you next time.
Yeah.
Thanks so much.
Thank
you, everybody.
Cheers.
Bye.